Okay. Wait a minute, sir. Naruh reference dulu.
So please start your video and the other group should be ready for asking questions. Okay, please start immediately because we don't have much time.
Share screen di. Okay, so good afternoon, Pak Dedi and friends. Today me and Andy will be presenting about the research that we have found earlier, which is vapor-based etang for sugar distinction. Next. Yeah, so it's a paper-based etang. First, they should fabricate the electrodes. So the electrodes are made of carbon, and here is how it looks. It has a 15 millimeter long and 10 millimeter wide, and it has 10 digits with 1 millimeter spacing. And the electrodes are printed on Whatman paper. And then they also print wax to make the reaction channel, which is in the middle. As you can see at figure C, this one, the middle part where there is the white one, that is the space where there is no wax. So they first made the template to be 3 millimeters wide, but after the whole electrodes and also the wax is heated above hot plate at 90 degrees Celsius, if I'm not mistaken, the channel radius become 2 millimeter. Yeah. Next. Next is the hydrogel. So they use hydrogel as like the membrane. The hydrogel, there are 3 types of hydrogel, which could be seen from the table here. Each of them contains PBA. Why they use PBA? Because they could bind into saccharides. And if we notice here, there are different amounts of contents inside the hydrogel. This is made because to produce different types of pattern, and the pattern will then be analyzed and pattern using the PCA or ITMAP. They are the machine learning system. And 0.6 microliters of hydrogel are placed at the start of the channel. Could you go to the previous slide? This one yang ada buletan itu di atas at figure C, ya, that one. And then they will wake through to the channel where there is no wax, the hydrophilic part. Go to the next slide. So they make 4 kinds of electrodes, 3 with the hydrogel, with this description here, and 1 with the bare electrode. So the signals that will be obtained from these different kinds of electrode will be processed through the machine learning, and they will provide the data needed. Next. The measurement will be done using electrical impedance spectroscopy. So they measure the impedance by applying 20 millivolt across the interdigitated electrodes in a frequency range from 0.1 to 10 megahertz with 5 points per decade. But they only took the data at the 100 kilohertz and 1 kilohertz point for sugar standards, because at that point the capacitance produced were larger. But for the apple juice, they took it from the 1 kilohertz point. and then the data will be analyzed using PCA and ITMAP. But at the paper, they only show the result from the ITMAP plotting. Next.
Okay. So for the so usually uh for Itang there will be 4 until 8 electrodes sensing uh sensing sensors, and for this it has four uh uh it has four sensors to differentiate glucose, sucrose, and fructose. And uh, it is it. So from the experiment uh, conducted, uh, which conclude that uh, it is better to it is better to uh, recalibrate uh, recalibrate the sensing the sensor to differentiate the sample, because if it does not uh, it can't effectively differentiate uh, differentiate samples. The left figure, it is IDMAP plot that is obtained uh, using the capacitance spectra of the four electrodes, uh, and so it immersed in the PBS solution, which is conclude with concentration of glucose, sucrose, and fructose. And the table on the right side is the concentration of each sugar and PBS buffer at pH 7.4. And uh, from this experiment, it shows they say it shows a great a great uh, a great result, which they want to calibration needed with different set of sensors, and they conclude that they want to uh, use apple juice samples of different what can you call different companies, which is Heinz Fruits, Tesco, Del Rivo, and Homestead. And since the Tesco and Del Rivo has uh quite close spec of sugar, and of sugar, so uh as you can see at the figure on the left, Tesco and Del Rivo is quite close because of the sugar and yeah, because of it has quite similar contents. And uh yeah, from this they get it is concluded that uh they should do calibration on each sensing to make a better result, to differentiate fructose, glucose, and sucrose, and to differentiate each of the uh, product. I think that's all. And this is the reference that we take.
Okay, thank you.
We took.
Group 2, please ask questions. And Lily and Ezra.
What is the function of hydrogel in the itang?
First, it could bind saccharides because the hydrogel consists of PBA. As we can see here, there's yang 5% AMPBA, 20% AMPBA, and the DMPBA. So we use the PBA of the hydrogel, which is the content of the hydrogel, to bind saccharides more. Kan glucose, fructose, and sucrose kan examples of saccharide. Jadi they want to bind the saccharides more, to produce promising results. That's the first one. The second one is to produce also different patterns. As we can see here, there is a varying amount of PBA. With the varying amount of PBA, there will also be some differences in capacitance results, and then the pattern will then be analyzed using the PCA and the ITMAP.
What is PBA, by the way? Chris?
Sebentar. Itu singkatan dari phenylboronic acid.
Phenylboronic acid.
Yeah.
Other questions from group 2? Anjali, maybe? No questions, Anjali? Anjali, are you there?
Yes, sir. Hello.
Okay. You don't want to ask questions?
No, for now.
No, okay. I want to ask question. First of all, the first slide. If you go to the first slide. Yeah, misspelling it. First slide, previous one. Title slide. Tonggue.
Typo. Sorry.
Okay, second slide. Yeah, so how many sensor arrays do they have here?
This is just an example of one.
This is one single sensor element, right? How many of them?
Four.
Four of them.
Yeah, the 3 with the hydrogel and 1 with the bare electrode. That's one whole itang system gitu.
So for each of the system, they drop the liquid into that hole in the C part.
The hydrophilic part.
So what will happen? The liquid will go downward.
Yeah, through capillary action kan. It is on paper. It will go through capillary action to the reaction zone, and then capacitance will be produced.
How do they pattern the carbon into the digitated electrodes?
I think they printed it. Yeah, they printed.
You are sure?
Yeah, they said were printed on Wattman paper.
How do they print it? Using what method? Screen printing or
wax printing?
Itu for the wax.
Yeah. For the digitated electrode, what methods do they use?
Screen printing.
Screen printing.
Yeah, the equipment is called DEK248 screen printer.
Do you think if we can do this in our lab using our stencil printing method?
If there's the screen printer.
we don't have machine. Yeah, we have to do it manually in our lab. We use stencil printing instead of screen printing.
But I think it is possible.
If you learn to do so, yes, from Elga.
Yeah, because yesterday Kak Elga had also shown us like how do we print the electrodes and the wax part.
Yeah, so now the problem is to get that 1 millimeter spacing, right?
Yes.
It's very small. Yeah, if we can get it with stencil printing, that will be best. Okay, so this is using carbon. Next slide, please. So the 3 is that one, 5 millimeter, 5%.
Yeah.
MPBA and 30% IMPBA and 100% DMAPBA.
Mhm.
Okay. I think that will be all from me for this group. Thank you, group one, for the presentation. Okay, then next one we will have group two, Ezra and Chris. Not Ezra and Chris, Ezra and Anjali. Okay, you may share your screen.
Okay. So I will start the presentation. Good morning, sir. Good morning, friends. Today, Ezra and I will present about microfluidic organs on chip. So first of all, what are organs on chip? They are microfluidic devices for culturing living cells in continuously perfused micrometer-sized chambers in order to model physiological functions of tissues and organs. The aim of organs on chip is not to mimic a full organism, but rather to synthesize the minimal functional units that recapitulate tissue and organ level functions. There are the simple chips that consist of only one type of tissue cultured, and there are more complex ones that consist of two or more microchannels connected by porous membranes lined on opposite sides by different cell types to recreate the interactions between different tissues. And next. So the fabrication of the microfluidic chip is basically same with the other microfluidic. So the first thing is that a thin uniform film of photosensitive materials is spin coated in the silicon wafer, silicon chip, and then it will overlaid with a photomask, and then after that it will given the ultraviolet um, light. So the one that is exposed to the UV, the materials that is exposed to the UV will dissolve in the developer solution. However, the one that is not will uh, be shaped like in the picture, like being in here. So after that, the mold is done. So they use PDMS to create the um, microfluidic. And then there are another type, which is a single channel microfluidic device that is also fabricated with the PDMS stamp with two inlets. So in this paper, they create a lot of designs of sometimes complicated designs for the microfluidic because the different organs have different designs like that. Next. So they also um, need to plant the cells inside the microfluidic. So they use laminar stream to plate the cells, or they also use ECM proteins. So in this picture, these are the examples of the um, organ on chips design. So in the A picture, the cell it used just a single cell type, which is cultured as a monolayer on the planar rigid. However, in the B picture, they use two different cells. And then in the third picture, this is the examples of microcontract printing of ECM in the linear pattern on a thin PDMS layer. So it they want to copy the cells of muscles, so to create an anisotropic muscle tissue layer. So when the part of the PDMS film is released from the substrate, the cells will be bent um, and the cells will be contract and create bend like in the sea. And this allowing the measure of the cell contraction force under the flow. Next. So the application of this microfluidic and the potential uses is like they can create drug development, and they also can modeling organs level of physiology, and also um, can predict the disease and can study the disease, can analyze um, some infection and disease in the cells. Next. So these are the examples of microfluidics that they already made. They already tested in the A picture is the liver on chip, and then the B is the kidney on chip, which in which human kidney proximal tubular epithelial cells are cultured on the top of the porous membrane that is separating by two different channels. And in B, they analyze the cellular transport of the cells. And then for C is they make
Hello, Ezra? Sound is cut. You cannot hear your sound. Angelique, can you?
Huh?
We cannot hear Ezra. Can you?
No, I cannot hear Ezra either.
Hello, Ezra, are you there? She seems to be disappeared as well.
Yeah. No, oh, she got disconnected, I think.
Perhaps you can. Yeah. Yes, she is.
hello.
Yes.
Yeah, I'm back again.
Okay.
I'm so sorry. And the D part is the breathing lung on chips. They replicate the lung function in the microfluidic chips. Next.
Okay, so next are the advantages and disadvantages of the microfluidic organs on chip. As for the advantage, it is easy to control the fluid flow because it is a microfluidic system, which in turn enhances the differentiation, function, and long-term survival of many cell types. And they also can carry out high-resolution imaging and determine where in the tissues to look, or it makes the visualization of process in living organs easier, as cell types of one tissue can be positioned precisely and consistently relative to those of another in a microfluidic chip. And it also has the ability to design synthetic culture systems in which many different control parameters can be varied independently. So there is more control to um, design the synthetic of um, systems systems on organs or tissues. Meanwhile, the disadvantages is most analytical approaches, such as mass spectroscopy, actually require larger experimental samples, so it is unsuitable for use with microfluidic system. And there are also some organ functions that are not yet ready to be modeled on chip. And the spatial heterogeneity found in larger 3D organoids or tissue sections is difficult to reproduce on a micro scale. Besides that, fabrication of microfluidic chip also requires specialized microengineering capabilities. And there is also the risk of bubbles in microfluidic channels, which are difficult to remove, and they may injure cells and hamper fabrication and control of the chips. And this is the reference that we took, and that is all from us. Thank you.
So this is basically already a review paper,
Um,
right?
sorry.
I think it is a review paper, not an original research article. Summarizing the whole thing. Okay, group 1. Please ask questions. Andy and Chris.
You mentioned earlier about the anisotropic muscle cells, ya. Can you explain more? Tadi aku gak gitu nangkep.
Yang mana ya itu ya?
In the slide yang ada sel yang gepeng itu.
Ya. Yeah, so they use the wait a minute. So they use like to plant the cells. They use methods like laminar stream to plate cells for the ECM proteins, and the um, the the cells is planted to the PDMS, and the cells that is picked is the muscle cells. So they covered the cells. Why they planted it in the PDMS cells is to create the anisotropic muscle tissue layer. That is why, because it's a muscle cell, so basically the muscle cells is working, contracting and relaxing. So when the PDMS is released, the PDMS film is released from the substrate, the muscle cell will contract. That is why it is like bending like that.
Oh, the bending is the contraction gitu.
Yeah, the bending is caused by the contraction of the muscle cell.
Okay.
Interesting. Andy, any questions from Andy?
Could you please go to the next slide? Again. Okay. So that explained reaction, where did it happen? Is it on the in the middle?
Everything is in the. I'm sorry. Which
I mean,
design do you
like
mean?
on figure C.
Okay, in
Or
figure
D?
C. Yeah, because they replicate the guts structure, so it looks like this. Anjali bisa tunjukin yang bentolan-bentolan itu. It really looks like gut kan, and they calculate the shear stress of the cells and also the strain. So the reactions happen in the middle, because the cells is also in the middle. But kan itu juga itu juga ada flow-nya. Jadi mereka ngukur berdasarkan flow and also the shear stress and the strain of the cells. Gitu.
Okay. Thank you.
Okay, I have one short question. What will be the trend for, I think this review is in 2014, yeah? If you look at the reference, can you go again to the last slide? Final slide, yeah. 2014, right? Now 7 years have passed. What is the current trend of organs on a chip? And what will be the future trend for the development of organ on a chip?
I think for the currently, there are and in the future, since this review, there has been more types of organs on chip, like more diseases that can be modeled and more systems. So I think that is the trend that will continue to that will continue in the future. So the currently unavailable system, like previously I mentioned that the disadvantage that there are some functions that are not ready to be modeled, and maybe in the future they can be developed in the microfluidic chip. Yeah, like that, because
Okay.
there are a lot of systems, right?
For example, can you mention some examples?
Yeah. In the current studies kan udah dibuat kayak misalkan low cost bioreactor for brain organoids, something like that. That is pendekatannya, the approaching of today's studies. I think it's more into cost, so low cost microfluidic organs on chip, and for the further development, as Anjali said, mentioned before, yeah, the pengembangannya lebih luas lagi
Lebih banyak yang bisa
dan lebih
dimodel.
detailed.
Okay. Do you think that we can play a role in the development of the organ on a chip in our lab?
Maybe for now, no, sir.
Not for now.
Besides, the cell is expensive juga.
So that we have bacteria in our lab.
Oh. So
I still said no, sir.
So for example, if
But maybe in the future we can.
in the near future, yeah, in next semester maybe. Biomedical instrumentation project.
Next three semesters, sir. after we done our thesis.
Interesting, yeah, to build a simple one, for example. I don't know what could be made, yeah, simple one. Maybe collaborate with other university which has the cell line, something like that. Okay, thank you, Group 2, Ezra and Anjali, for the presentation.